virology lecture, unlike other lectures, I will read out all the points and then explain, not like other lectures. Because these are the general properties, okay? I want you to know each and every property rather than if I tell you like a concept, then something might be misrealized. So I will be reading each and every point, and then actually this lecture is in a very simple words. It has been put in a very simple words. Even if you read it, you will know it. So that's the reason I will read it points wise and then explain it. If somewhere something you don't understand, you can stop. Okay? After taking bacteria, I'm sure this will be very easy. Now, please tell me what you know about the virus. The most important virus you have taken already. HIV. What do you know about the virus?
It's not a bacteria.
Put this. Okay. Yes. And very
It has
important.
either DNA or RNA.
Good.
That's living.
How do we measure the size of bacteria? Units. Huh? Nanometer, you said? You said? Bacteria or virus? Bacteria, I asked. Bacteria. How do you measure?
Micrometer.
Micrometer. Now, as you all said, that is what a virus is. All those points. What? First, we will start with the terminology. What do you mean by a virus? It is actually a Latin word which means poison. Back in 18th century, even before that, there were some tiny particles. Just imagine bacteria itself is a tiny thing, isn't it? Virus is more tinier than that. So somehow this scientists, they saw something, they made it out that it is not bacteria, it is something else. And this is causing disease. This is causing some problem with the human body. So vaguely, they named it as poison. Because what is poison? It harms human body, isn't it? Poison, it is harmful. So as this particle was showing harmful effects, they named it vaguely as poison. Poison is virus in Latin, okay? So this is how the name virus was acquired by this community basically. Now, I told you about the size. Talking about the structure, we will deal with structure in detail. This is all summary, okay? Just points. Everything will be dealt in detail accordingly in different lectures. Like replication, you will see in detail in replication. The structural thing you will see here in this lecture, okay? So basically, to start with, these viruses are enveloped, most of them. Few are non-enveloped, okay? Now, what is this envelope? You compare everything with bacteria. That is the reason this module has been placed like this, okay? Because you have taken bacteria just before this. So now you will compare everything of this virus with bacteria. What was the envelope of bacteria? Capsule, cell wall. These are all coverings of bacteria, isn't it? So here it has only envelope, which is a lipid bilayer. Okay? Lipid, it contains lipids, proteins, glycoproteins, many things. But the basic component is lipid bilayer. Alright? That is the envelope, outermost membrane of the virus. Now few viruses, they lack this envelope. Not necessarily that all organisms should have cell wall, isn't it? I mean in bacterial language I am saying. So here also, not all viruses will have an envelope. Few are naked virus. Now, what do you mean by naked virus? Without clothes? So what will be inside it? Capsid. Okay? I will show all the diagrams. Okay? You will deal with them in detail. Now the innermost part is capsid. So few viruses, they have only this capsid with the genome inside. That is the entire body of the virus. whereas few are covered with the envelope. Got it? Few are covered, few are not covered or naked. Now, here, just imagine, do you know, have you any time, I'm not sure how much you are interested in cars, but do you know how car is made, how it is produced finally? Car, vehicles, no? Do you know this, that I mean, first the parts are made, they are imported from a different place, they come to a different place, they get packaged. Do you know this? Everyone? Only few know. Okay. So it is just like those vehicles. Unlike bacteria, virus does not grow. It does not grow. It is a basically, they say it is not a living thing. Okay, because it does not grow like bacteria. Now, how is it produced? Different parts are made. Okay, then they are assembled. You have seen in HIV replication, isn't it? You remember through budding it comes out after assembly, viral assembly we say. It's just like that. Different parts are made. First the genome is made, capsid is made, envelope is made, proteins are made in different parts of the cell. They come at a point, all this gets assembled. Okay, all this gets assembled and then the envelope is formed. Only in envelope virus, not in every one. Envelope is formed, then the virus is released. This is how virus is made. How bacteria is made, tell me. How they are going to use binary fission. Okay, they multiply, isn't it? Just like a living cell, they multiply. Whereas virus, no, it is not a living thing, so it does not multiply. Different parts are made and then they are made to assemble in a particular corner of a cell. Then they are assembled in a envelope and then they are released. So this is another difference between bacteria and virus. Clear? I'm sure you remember this. It doesn't write Pura. It's okay. Okay. I will just hold it to show you. Bacterial growth curve. You know, what is it? How many phases you see?
4 phases.
What are they? Good. You know, there are 4 phases and bacteria first it starts with a tiny thing. It multiplies, it undergoes all these phases and in all this entire growth curve, the bacteria will not lose its infectivity, isn't it? It is, if it is a pathogen, if it starts as a pathogen, it is pathogenic only throughout the growth curve, isn't it? The difference between virus is not like that. Because as it is a non-living organism, the growth curve of virus something it looks like this. Now what is this? First, when the process is starting, when the growth curve of virus is starting, it starts with an eclipse period. Now what is this eclipse period? This eclipse period is nothing but, here the viral particles are being made. Okay, different particles are being made. And another important difference, I told you in the entire bacterial curve, whenever it is dividing, the infectivity is there. Whereas it is not the same with the virus. It is not infective when it is in eclipse period. Neither can you detect virus in the human body in this phase. Clear? This is a major difference. Did you understand? Eclipse period. Again. Eclipse period is nothing but the start of the viral growth curve. Okay. In this, different parts of virus is being made. So, virus is not infective in this stage. Neither it will have its infectivity, nor it is detectable in human body if it is in this phase. Okay. This is one of the differences. Now, once the eclipse period is passed down, after some time, all the viral particles, genome, everything is made. Virus is assembled, it is formed. Then it will form a burst size or yield phase. Okay? Yield phase means where the virus is ready and it is detectable and it is infected as well. Okay, all of a sudden everything changes. Unlike bacteria it is proportionate in all throughout the life cycle. It's not here like this. It has no infectivity here, no detection in human body, nothing. Once it is assembled, suddenly, can you see, this is gradual, this is sudden. Because all the viruses are made here, they are assembled, and suddenly the burst size increases. What is the burst size? The quantity of virus formed per cell. Okay? Quantity of virus, number of virus particles. Did you understand whatever I said? It's very important for you to understand, okay, the growth pattern and the difference. And the growth pattern and the differences. Please. Okay. Everyone? Quantity, this burst, here there is a word you can see, burst size or yield phase. It is nothing but the quantity of virus produced per cell. There are many cells, right? The virus is multiplying many cells. So per cell, how many virus have been produced is the burst size. And this entire yield phase and eclipse period, combining these two phases, it is known as latent phase. Okay, you have another graph showing that. Anyone don't follow, please tell. This is a little bit of history when the first virus was discovered. Do you have any idea about this? The first virus, yellow fever virus? No? It was back in 1901. The first virus discovered was yellow fever virus, and it was named yellow fever because it caused some kind of, even now we call it as yellow fever, but back then they did not have much knowledge about it, so it presented with fever and yellowish discoloration of the body, including the mucous membranes. So they named it as yellow fever, and the virus as yellow fever virus. Okay, followed by rabies. We all know what is rabies, isn't it? Isn't it? Variola, chicken leukemia, polio, sarcoma, bacteriophages, and influenza. Now talking a little bit about more of the properties. Filterable agents. What do you understand by this term filterable? In our homes we have filters, right? If you see the advertisement or if you read the filter, the carton which comes with it or the booklet which comes with it, bacterial filters there are. Have you ever seen, noticed? We must. Anyway, so they are all bacterial filters. They have been designed, in fact back in 18th, 19th century it was totally disaster. The filters were quite different. Only it could capture the larger organisms, even not all the bacteria, few bacteria, but most of the bacteria. But virus, no. So those times the filters used for bacteria, they tested for these organisms. But they saw that these organisms are able to get through these filters, through the bacterial filters where the bacteria is trapped, but the virus is coming out. Okay? So one of the characteristics is it is filterable agents. That means it denotes the size of virus in short. Okay? Another thing, I told you about this. They do not grow. So basically, as the bacteria grows, it liberates energy, isn't it? There is a lot of energy and many things which gives bacteria energy to grow. There's a lot of energy involved. You have read, you have taken that entire life cycle, isn't it? In virus, there's nothing like that because it is not growing. It is completely dependent on the host cell, virus, unlike bacteria. Okay, so it will completely utilize the host cell energy to get created. The different parts have been created and then assembled. So this energy is taken completely from the host cell. Basically, it is a parasite. You understand what is parasite, right? It completely takes up the host energy for its growth and survival. It will not grow by itself like bacteria. Do not produce or store energy. I told you it is dependent on host cell energy. Are not functionally outside the cell. These viruses are obligate intracellular parasites. Okay? Bacteria are intracellular or extra?
Intra, both. Intra and extra.
Few are intra, many are extracellular actually. Few are intracellular. Whereas virus, if it is extracellular, then it is of no importance for us. Okay? Because because they are active only inside the cell. Now tell me the reason for it. I just told you. Why?
They don't grow.
Yes, but what exactly is it?
Depends on the host.
Exactly, because they are completely dependent on host. Completely. If it is outside the host cell, it is gone. So that's why they are obligate intracellular parasites. The viruses are. Okay? Talking about presence of these viruses in the environment. Bacteria are also seen in the environment everywhere, but viruses are little bit more seen in environment everywhere. Everywhere it is seen. Not just it is seen, but it will infect everything. Unlike bacteria, even bacteria also is infected by virus. You know that? Yeah. So basically, it is not going to spare anyone. This virus. So it is known as ubiquitous in origin. I mean, it is found everywhere. It will infect anything which comes in its way. Talking about size, they are very tiny. We know that tinier than bacteria. The size varies from 18 nanometers to 300 nanometers. But there is another virus, Mimi virus, I think. Yes, Mimi virus, which is little larger than these viruses. It is around 400 nanometers. So still scientists don't know whether to put it in virus or in bacteria. But still it is in virus. It is not considered much of a virus because of its size. Rest all the characteristics are like virus only. Other than that, we put the size range as 18 nanometers to 300 nanometers. You all know about its genetic material. What it is? Both?
Both DNA and RNA.
Either this or that, isn't it? Talking about classification, as we know that this is not a living organism. Bacteria, you have taken bacterial classification or it is yet to come? Yet to come, right? You will know. Bacteria has a very systematic classification because bacteria has got many growth factors. So depending on these properties, it has a proper classification. Whereas virus is not like that. Here, you all might be knowing Carl Linnaeus. Bacteria are named by him back in 18th century. Carl Linnaeus. So it is known as linomial classification. If you want, you can remember, otherwise no need, but I'm just telling you, okay? But whereas he couldn't name virus because he did not understand on what basis he should name the virus, because there is nothing similar like bacteria. They are not growing, they are not giving out any properties to be classified. They don't have a particular ancestor that they can be classified, isn't it? They are all ubiquitous in origin. So he couldn't name, he just gave up, and so he couldn't name virus. He just named bacteria. He gave a long elaborated classification of bacteria and he ditched viruses. So basically later on viruses were named somehow to the most highest order is family. That's it. Okay? I mean, there is much of classification because you have not taken classification of bacteria, maybe you are not able to understand. There are many classes, okay? Subclasses, types, subtypes, many classification, you have a lot of criteria. Whereas that criteria is not seen in viruses. I know it's a little boring, but the general is like that only. So, what do you think how this virus might have been classified? Either depending on the place where it was first seen, or depending on the disease, or depending on any of its morphological features. This is how they have taken a name and they have classified each virus. For example, picorna virus. Picorna means it is kind of a small genome. Okay, so they named it as picorna virus. Talking about diseases, adenovirus is named because it attacks adenoids. Like that they named. Okay, and there are many viruses. Many viruses you will see with the place names. Coxsackie. These are all in United States, some of the other place. There are many viruses with the place names attached because these viruses were isolated in those places. This is how they were named and classified. Only these many categories are there for classification of virus. Even not all the viruses, only a few are classified like this. Other than that, a few are just left with individual name, that's it. Apart from that, depending on the type of genome which is present in the virus, there is another classification which is Baltimore classification. We all know either it has a DNA or RNA, okay? Some viruses, they have double-stranded, some have single-stranded, some have RNA which is positive sense, some have RNA which is negative sense. I hope you remember what is positive sense and negative sense RNA. No? So what is it? You are right. What does it do? You are right. It is like identical to mRNA. You are right. But what is that modification? You got the concept, but tell me the name of the process. That is HIV. Reverse transcriptase is only exclusive for HIV, not for any other virus. What is positive sense? What is negative sense? You have taken this even before our immunology lectures also, you have taken in your other blocks, I think. In which block did you take this genetics? Premature you took, right? Which block was that?
Genetics.
Genetics is the block. So, you don't remember? No? Okay. Let me brush you up. Positive senses, as she said, it is like mRNA. Now, what this mRNA will do? Directly, when it enters the host cell, directly translates into proteins. What does the negative sense does? Once it enters the cell, first it has to be transcribed into mRNA. Okay? Because that negative sense RNA is not capable of producing proteins. I mean, not capable of translation. Okay? So first it has to be transcribed into mRNA, then it will be, and it has to carry its own RNA polymerase, the enzyme, through which it will become mRNA, and then it will be translated into proteins. Okay? So this is the difference between positive sense, negative sense, which you have to know for sure. Okay? Okay? I will repeat in the next lecture. You have a detailed thing in replication. Alright? So, this is positive and negative sense. So, depending on the type of genome present, it has been classified by Baltimore. Another type of classification is, I told you that a few are enveloped, few are non-enveloped. Now, this is a classification completely depending on the envelope and naked viruses. First for DNA virus, if it is enveloped, there are a few examples like pox, herpes, hepatitis. You have to know these examples. You have to mug it up, okay? And naked capsid virus you can say, or naked virus. These are the examples of native viruses. Now talking about RNA virus, again these RNA viruses are classified into positive sense RNA, then again into naked envelope. Then negative sense RNA, again negative sense RNA are only enveloped viruses, and you need to know this examples. Few viruses, they have both positive and negative sense. They are known as ambisense. Okay? Where they have one end is positive, other end is negative. So they are known as ambisense genome. Now, positive RNA via DNA. Which virus is this? RNA from DNA. Reverse transcriptase. HIV. So that is one specific virus classified completely in a different category. Here, this is the structure of an enveloped virus. Can you see a bilayer here? The outermost layer is a lipid bilayer. Alright. Now this has come from host cell, isn't it? Because virus, where is it made?
In the host.
Inside the cell. So these are from the cell components. Okay? It is made up of basically lipid. Apart from lipid, it has all these, what are these spike-like structures?
Proteins.
Proteins, glycoproteins, hemagglutinin, what are all this, I'll tell you. Basically, they have a lot of proteins inside it. These are different kinds of proteins. Now, coming a little bit inside, can you see the structure? What is it?
Capsid.
It is a tough, hard structure, protein shell basically, which is known as capsid. What is a capsid? Capsid is nothing but it is a protective layer of the genome. It protects the genome. Genome is the most important part of virus, isn't it? If genome is removed, there is nothing. It's like an empty box. As if you have taken soul out of human body. If genome is gone, everything is gone. Now, so this is genome of course. Whether it is RNA, DNA, there are many enzymes, genomic enzymes, proteins, structural, non-structural proteins present inside it. So this is a complete structure of a virion. Is it clear? It's easy. There's nothing much in this. Unlike bacterial cell wall, the huge thing. There's nothing in virus. Okay? It's just this one. Proteins, as I've told you about capsid, let me show you the next slide. Talking about a little bit details of these structures, what I have told you in the diagram. Capsid, I told you, it is basically a hard protein shell which surrounds the genome. What is nucleocapsid? Another terminology, nucleocapsid. Where is it? Okay, here. When this capsid put together with the nucleic acid inside it, with the enzymes and proteins inside it, it is called as nucleocapsid core. Okay? It is not any separate structure, but it has been named like that. This is capsid with the nucleic acid inside it, with the proteins and enzymes put together is known as nucleocapsid. Envelope we all know. Virion. What is virion? What is virion? Till now I was talking about virion only. What is virion? Infectious virus particle is nothing but virion. Okay? It's nothing different. Now, these viruses, I told you there are a lot of proteins inside the viruses. So these proteins are divided into two types. One is structural proteins. The second one is non-structural proteins. Now please, little bit use your brain and tell me what might be the use of structural proteins. Structure.
Making the particles.
Replication. You said replication. That is non-structural protein. Structural protein. Is it used in something?
Catch
Actually, it is used in all the making of structures like capsid, envelope, all these structures because they are called structural proteins. Okay, these proteins are utilized in making the structures of virus. Now, what will be the structures of virus after seeing the picture? Envelope is a structure, capsid is a structure, isn't it? In these, the structural proteins they contribute. Okay, whereas non-structural proteins, as she said, they help in replication. Okay, these are the two types of proteins. Interaction with host cell membranes to form an envelope. Yes, it is also one of the functions, which we will see in detail. Now, these structural proteins, what are they going to do? As I told you, they have formed the structure, isn't it? Maybe you will understand this better. But no, you have taken HIV, so you will be understanding this also. These structural proteins, as the structures are formed from these proteins only, so these are very helpful in different stages of the virus life cycle. What are those different stages? Tell me the replication stages, do you remember? Right, binding, attachment, uncoating, fusion, replication, assemble, right. Brilliant, very good. So basically in many of the functions these structural proteins will contribute because all these proteins are present in the capsid and envelope. Okay, so basically it will help in binding of the because it is present in cell receptors also, the receptors through which it binds. So it helps in binding. After it gets inside the cell, it helps in uncoating. How it will help? It will break down itself because capsid is made up of these structural proteins only. That is how it helps in uncoating of the genome. Fusion of envelope with the cell membranes. Transport of the genome to appropriate site. Why does it say appropriate site? What are the sites according to you? Because you have taken this in pre-med. Why does it say site? Where does this replication occur? Only in viruses, the DNA virus replication is different and RNA virus replication is different. The major difference is site, where it occurs. Okay, so one will occur in nucleus, that is DNA viruses. The replication takes place only in the nucleus, with some exceptions. You will know the exceptions, but majority of them it will take place in nucleus. Whereas RNA viruses, the replication will take place in cytoplasm, with some exceptions. Alright, so that's why it says transport of genome to the appropriate site. So if it is DNA virus, where will it go?
Nucleus.
If it is RNA?
Cytoplasm.
These are the same points repeated. You want me to repeat it? These are the same what I have been telling you, repeated. The adhesion and how it protects the genome, how it carries. I have been telling you this. Now, I have explained you this also. Talking about genome. As we all know, it is RNA or DNA. Okay, DNA may be single-stranded or double-stranded. RNA also it might be single-stranded and positive sense and negative sense. Most important in RNA is this: whether it is directly mRNA or indirectly mRNA. Positive sense, what it does? Direct mRNA. Negative sense? That is, I just told like that. Don't write like that in the exam. Direct mRNA and indirect. For your understanding, I just said like that. Okay. Now tell me what is negative sense RNA in medical terminology. Huh?
Should it transcript it to mRNA?
Right.
And
it
then?
has to undergo transcription. It has to carry its own RNA polymerase. It has to undergo transcription,
And then?
form mRNA, then it will be
Translation.
translated into protein. Okay, that is negative sense. Positive senses, it directly translates. Yes, I told you they have to, this negative sense, they have to carry their own RNA polymerase. Why?
Because they have to translate the RNA into
You are right. But why DNA viruses are not carrying anything with them? Why only RNA viruses are there? Because the enzyme needed for this mRNA transcription, it is not available naturally in the host cell. Okay? Whether it is human host cell, animal host cell, whichever the host cell is, it will naturally not carry this enzyme, which RNA wants to use. Not wants to use, it has to use if it has to become mRNA. So, the condition is it carries its own enzyme with it, then only it can transcribe first and then translate it. This is an important thing, important question, why it has to carry its own RNA polymerase. Okay, please try to remember, because host cell will not naturally have this enzyme in it. That's why it has to carry. Now, talking about DNA and RNA virus, I don't know how much you are interested in medical history, I'm not sure, but if you go through, sometimes if you have a little bit of time and if you are interested, if you go through, how many ever pandemics or epidemics have been occurring, this viral epidemics, you will see most of them were RNA viruses. Why? Because these RNA viruses, I mean the RNA, the genome RNA, it lacks proofreading capability. What do you mean by proofreading? For example, today you are taking a lecture. You read it. Fine. Tomorrow you have PI. You will check. If somebody has a habit of writing, I have that habit. I don't know if anyone has habit of writing. Before going to take an exam, you will proofread yourself. You will check for any mistakes. Double checking. Isn't it? We do it. You don't? Every student does it. We want to check for our mistakes. So that reading, again and again, reading and correcting yourself is proofreading. Okay? So this DNA has this proofreading capacity. It does for itself. That's why the rate of mutation is very less, because it is always checking its mistake. When you check yourself so much, you will make less mistakes, isn't it? That's why the mutation rate is very less in DNA. Whereas RNA takes it like, yeah, it's okay. It will not proofread itself. So the rate of mutation is high. That's why the pandemics what you have seen till now, the epidemics what you have seen till now, majority of them are RNA viruses because of high rates of mutation. Every time they change one spike protein, one glycoprotein, they mutate and they produce a new disease. It's like that. Have you heard about H1N1? Influenza, H1N1. It was recent only. H1N1. It was an epidemic a few years back. Anyway, so what is this H and N? H stands for hemagglutinin, okay? N stands for neuraminidase. You know what are these H and N? These spike projections are H and N, okay? The glycoproteins present on the wall of the virus. These are hemagglutinin and neuraminidase, okay? So the mutations occur in these spike proteins. When there is some mutation, the virus will change its entire system is changed and it will produce a new disease. This is how it happens. Okay, so basically back to our point, proofreading. Now you understood what is proofreading? Hope it is clear to you. Very good. So this is the reason why mutation rate is higher in RNA viruses than in DNA. This is nothing but Baltimore classification what we have seen. you taken lecture on bacterial spores? Spores.
Yes.
Yes. What are the spores? How do they differ from bacterial cell? Huh? Sorry? Inactive you're saying. Anything else? Is there an active? Anything else? Huh? Multi? Huh? Yes. And basically they are, what I wanted from you is, they are very hard and tough shell in which bacteria lives in an inactivated form, and this survive all the drying and heat and harsh environment, isn't it? Spores are that only. It will survive. In the same condition, if bacterial vegetative cell is there, it will be gone. It will be dead. But whereas in spore form, it is alive because the spore wall is very thick and it is resistant to harsh environments. Same thing with capsid. Capsid is nothing but tough, hard protein shell, okay, with enclosed genome. Alright. Now this capsid is very hard and tough that it survives the gastric acid. Our gastric acid is highly acidic, isn't it? Because it is an acid. Whereas many viruses, bacteria, they reach gastric acid and they are killed there. Whereas this capsid survives gastric acid and bile. Okay? Basically it is a tough structure. So this gives you a clue that all the gastroenteritis causing viruses are naked viruses, because envelope will not survive this acidic environment. Once it goes there, the envelope is gone, virus is gone. Whereas these naked viruses, they survive this acidic environment and they cause the disease. That's why the naked viruses are mostly seen in infections of gastrointestinal tract. Now you know the reason why you see naked viruses in GIT. Why?
Because they survive the acid.
They survive the acid because of the toughness of the capsid. Okay? Whereas envelope, no, envelope will be disintegrated in the acidic environment. Okay? Now, so can you name some other system where you think envelope will survive? As I told you about the capsid, where do you think the envelope will survive? Basically, in the other systems where there is no acid found, whether it is urinary system, whether not much in urine, virus is not much in urine, but respiratory, yes. And this enveloped virus, they need a wet environment. Okay, so in our respiration, we have a lot of mucus, isn't it? So basically, in these kind of environment where there is humidity around, these enveloped viruses are seen surviving and causing diseases. Now talking a little bit about envelope, as I've told you already, this lipids, proteins, glycoproteins, and okay, how this envelope, how virus gets this envelope? After getting, after all the parts are assembled, everything happens. In the last step, if you remember, which we will get in next lecture, in the last step, after assembly, the host cell products will form the envelope. It will surround the capsid or the virion, and then the envelope is produced and it gets cut from the host cell. Do you remember the diagram? Yeah? Like a bubble, it comes out. That is how the envelope is formed, and it comes out through budding. Either through budding or through cell lysis. Cell lysis means? Cell breaks open to release all the viruses inside it. So it's not always cell lysis, sometimes it is budding. So cell need not die every time. Okay? Sometimes it comes out via budding. Here. These are the different viral structures after replication and all, they are formed, isn't it? Here you see genome, you see capsid, these are different proteins which are present inside the virus. They come here at a certain point of the cell. This is the host cell membrane. Okay, it takes up little part of this host cell. It takes inside it like this, and then it forms its envelope. Okay, now you got it, how envelope is formed from the host cell. You remember this? Cell mediated immunity. Okay, what do you think about that picture? Forget about immunity. What do you think is happening there? Just looking at the picture, you can say something, isn't it? What is happening there? What do you see in first picture? What do you see in second picture? They come together. Yes, you are right. Basically, this is seen only in viruses. You don't see this in bacteria. No. Not at least in majority of the bacteria, but in viruses you see this. And what happens? In pathogenicity you will get this again. Here, these are all uninfected host cells. Uninfected, good, healthy host cells. Once the viral genome is introduced in a cell, that means one cell is infected. Now here, if one cell is infected, take this cell only, okay? Within this cell, the entire replication process happens. Now, after happening, the entire process, after virus assembly, the complete virion is formed, okay? Infectious virion is formed. What this virus is going to do? Will it stay here only? No, it is going to attack another cell adjacent to it. It is going to attack all the cells adjacent to it, first by attachment. You know the process, how it enters the cell, isn't it? First it attaches via receptors. So via receptors it will attach. It will cover all the uninfected cells beside it. Okay? Now, after infecting that, these cells will fuse together. This is the specific property seen with viruses only. Okay? And when the cells fuse together, they are all infected. And this is called as syncytium formation. Okay? You understand what is syncytium? And this is called as cytopathogenic effect. CPE. Okay? Cytopathogenic effect. There are many types of cytopathogenic effects. This is one of them. Okay? Syncytium formation. There are many other, like ballooning of the cell is there. Many other things are there. But this is one of them. Syncytium formation. When you see this picture, it should come to your mind, it is syncytium. What is syncytium?
Fused together.
Fusion of uninfected, infected cells. Ultimately, everything will become infected because of the production of virus. See the structure and tell me what you understand. That is the structure of capsid. What do you think it is? All the time we were saying capsid, 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 isn't it? I told you this capsid is made from proteins, different structural proteins. Now how this capsid is made? First, if you see that structure like a triangle, first it is made into these small, what it is called? Capsomeres. No, there is another name for this. Protomers. That single unit is known as a protomer. Okay, first it is formed into this single unit, protomer. Now these protomers will come together, they attach to themselves and they form a capsomere. Alright, this is a capsomere. Now many capsomeres come and form this pentavalent or hexavalent structures. Okay, now this form a capsid. So basically capsid, unlike spore, is not just a shell. It is a symmetrically made structure. Do you understand what is symmetrically made? These are symmetrically made, isn't it? So that's why it gives more toughness and more strength to the capsid. The reason is because of this structure, okay? And this is how a capsid is formed. There is a structural difference of spore and a capsid. This makes it more tough than bacterial form. And this also, another point, this also serves as PAM. What is PAM? Pathogen associated molecular pattern. You remember PAMs, right? PAMs are nothing but the attachment sites from where the virus recognizes the cell and they interact and then they infect. So this capsid, because of this structural, specific structure, it forms PAM, okay? A mode of infection or source of infection. Those have been told, but this one here. In this is what we are talking about, naked structure here, capsid. Now when the capsid is released, usually the cell lysis happens, okay? Cell is broken and then number of virion are released, that is naked viruses. Now, as soon as they are released, they are exposed to antibodies. Whereas in envelope, cell-mediated immunity is enough. I mean, no need of neutralizing antibodies, not needed much in envelope. Whereas in capsid, as it releases by the cell lysis, they are exposed to antibodies. Now, which antibody is going to help with this? Neutralizing antibodies. Neutralizing antibodies are enough. The primary antibodies, they are enough to contain this infection. This is the reason why in viruses, CMI plays an important role than what is the other immunity? Huh? Huh? Innate is only CMI, no? What is the other immunity? Humoral immunity. Humoral, what happens? You will see all kinds of antibodies coming, attacking, isn't it? Here in virus, you will not see that. CMI is enough because this is within the cell. Cell-mediated immunity is enough to contain that infection. Only in capsid, I mean only naked viruses, that too, a little bit of neutralizing antibody is used here to contain the infection. Otherwise, for viruses, CMI is enough to contain the infection. What do you mean by tropism? Tropism of virus? Tropism of anything. What do you mean by tropism? Cell tropism, tissue tropism. No? Selectivity. Have you heard neurotropic? Lymphotropic? No? There are some viruses, we will not talk generally, we will just talk about viruses. There are certain viruses which show tropism, I mean affection towards a particular thing, okay? Neurotropic virus, for example, it will go and attack only the neural tissue. Even if something else comes in between, it will say no. It wants only neural tissue. Some other lymphotropic viruses, it will go only and attack lymphocytes. They are not going to attack something else. So this is called as tissue tropism, okay? Because it will have affinity towards that particular organ or system. Got it? Tropism. Envelopes are generally round in shape, but only two exceptions. Not just two, many are there, but pathogenically, the viruses what we see in everyday life, two more important viruses are rabies virus. If you see the structure of rabies, wherever you see a virus diagram, how do you see it? You see it like this, isn't it? Or like this. The only virus which is very different is this, the rabies virus. It is like a bullet. Okay, so basically these are small exceptions, nothing much about it. Viral glycoproteins, I told about it. The spike proteins on the envelope, they are nothing but the glycoproteins. These are different examples of envelope virus. Now talking about hemagglutinin, the spikes, you remember on the spiky projections on the envelope of the virus, one of them is hemagglutinin. Hemagglutinin is nothing but, it is a viral associated protein with a special characteristic. You must tell me that you have taken hemagglutination. What do you mean by hemagglutination? You have taken the lab on it. Forget about lab. You tell me what is hemagglutination. You are right. Who told? You told? You told? Someone told. What is it? Right. These glycoproteins, some of the glycoproteins on the envelope of the virus, they have the ability to clot RBCs. Okay? These are known as hemagglutination. Okay? The process is hemagglutination. The spike proteins, they are known as hemagglutinin. Okay? The proteins which have ability to clot RBCs, they are known as hemagglutininin. There are many spike proteins. I told you another example is neuraminidase, but we are talking about hemagglutininin. Few certain viruses which have this hemagglutininin are these viruses: influenza, measles, parainfluenza, and the variations, the mutations occur in these spike proteins only. When the mutation occurs in these spike proteins, the diseases, the virus changes, and the diseases are also changing because of these proteins. There is a diagrammatic representation of viral hemagglutination. This forms the basis of many tests actually. Hemagglutination we have taken last year, right? Remember? In serology we have taken this. That finishes viral structure.